Good morning, everyone. Welcome to the second part of our webinar, which is on extractable and the leachable. In this webinar, we are going to discuss about the validation and the study sample analysis. Allow me to introduce myself, Milan Modi, and I'm working as a Director of Operation at Zenovel Pharma Services. And here I'm going to present this second part of extractable and leachable on validation and the study sample analysis along with my colleague Mr. Ketal Prajapati. I request him to introduce himself.
Good morning, everyone. Warm welcome to all of you. My name is Ketal Prajapati. I am working as an Associate Director for the GXP services at Zenovel. I am having more than 20 years of experience in the field of preclinical, clinical research and analytical support. So let's start our insightful discussion on extractable and leachable. So today we are going to cover a validation and study sample analysis part. Thank you, Milan.
Thank you. So what we understood, what are the extractable and the leachables? Those components which are undesirable and which are coming from the packaging material, primary or the secondary packaging material, and those might be harmful to the human body, and those can be found in the dose. So it is very important to identify them and see their toxicity level. So what we understood so far, this is the recap. We understood the objective to perform the extractable and the leachable studies, the introduction and discussion about the sources from where the extractable and leachables come from, typical extractable and leachable study workflow, steps for the extraction studies, simulated versus controlled extraction studies, solvent selection, extraction conditions, and analytical technique which are used for the extractable and the leachables. So now from this webinar, we will get idea about the validation part, what are the parameters to be validated and how to perform the study of extractable and the leachable, which are the key component of the study.
Okay, let's start from the method development and validation. So for the leachable study, why we have to perform the method validation? To develop a method which is a sensitive method. So why we require the sensitive method? To achieve the LLOQ at or below the AET value. And the validation is nothing but an ICH acceptance criteria that we have to follow before the performance of the study sample analysis. Okay, so development of the leachable and its analysis includes analysis of VOCs, SVOCs, NVOCs, and elemental impurity analysis, inorganic ions, and low molecular weight organic acids identification. So for estimation of all these kind of impurities, ready to use reference standards are also available in the market for the evaluation of such kind of impurities. So if we see the requirement for the method validation, it is an US FDA expectation to have a validated analytical method for the estimation of the leachables, and for the it is also useful for the reliable detection, identification and qualification of the leachables. So FDA has guidance for the container closure system for the packaging for the human drugs and biologics, and for the method validation we have to follow ICH Q2R1, and for the assessment of the extractables USP 1663 chapter is available, and 1664 for the estimation of the leachables.
All right. So these are the guidelines which says that method development and validation is required. So can you focus more on the which parameter to be validated? Is it same as the bioanalysis, or they have some different criteria or the parameter to be validated?
If we see, moreover, the validation criteria for all the kind of analysis, if, for example, if it is a bioanalytical or the analytical, few parameters are almost same. So let's start with the first parameter, specificity and selectivity. So specificity is useful to distinguish the leachable from the drug product components, for example, active ingredients or excipients, and also it is useful to assess the interference from the matrix. The next is LOD, limit of detection. It is useful to determine the lowest concentration at which the leachable can be reliably detected, and also important for the compounds expected at a trace level. The next parameter is LLOQ. So generally, when we are selecting our LLOQ, we have to keep in mind the SAT and AET value. So SAT is the safety concern threshold, whereas AET is the analytical evolution threshold. So determine the lowest concentration that can be quantitatively measured with acceptable precision and accuracy. So it should be meet the safety threshold requirement that we have already discussed, SET and AET value. The next is linearity. To confirm the method response is linear over the relevant concentration range. So typically the linearity is tested across the range from the LLOQ to the level above the expected maximum concentration of the leachable. The next is recovery. It is also called as accuracy. So assess the recovery of known quantities of the leachable spike into the drug product matrix. It is helpful to ensure no matrix suppression or the enhancement, and how much recovery, how much leachable we can extract from the particular container closure system. The next is precision. It is nothing but a repeatability and intermediate precision. So repeatability in terms of within run accuracy, and intermediate precision, it is between run analysis or the instrument consistency. The next is to check the robustness. So evaluate methods resilience to small deliverables variables. For example, small change in the temperature, flow rate, mobile phase condition, pH, little change in the extraction condition can be evaluated as a robustness. So important to show the method is stable under the routine conditions. So it can help us to check the day-to-day variability, whether it is impacting the drug's concentration or leachables concentrations. The next is, and most important parameter, is the stability of analyte. So check the stability of the leachable in the matrix under the storage and analysis conditions. So additionally, we can also consider a system suitability during the method validation. It will help us to ensure the system is performing properly before and during the analysis. The next is matrix effect. So sometimes when we are using a complex matrices, then it is required to evaluate the matrix effect. For example, protein-based biologics. Then it is helpful to evaluate matrix suppression or enhancement. And if it is a possibility of any kind of matrix suppression or the enhancement, it should be a case of change in the extraction procedure.
All right. So thank you so much for describing all the validation parameters. I request you to please summarize all the validation parameter with its relevance to the study content.
Here the tabulated format of the validation parameter with respect to relevance of the leachable studies is provided. So if we see the specificity, it ensures the detection in the complex drug matrices, LOD, LOQ to meet the AET and SET levels, linearity for the accurate quantification over the expected range, accuracy to confirm the recovery of the spike leachables, precision is for the reproducibility within and between runs, robustness, the tolerance to a minor method changes, and analyze stability to ensure the reliability of the results during the sample handling.
So method validation is very much clear. So we would like to discuss more about the study part. So can you describe what are the major component of the study for the extractable and the leachable, and what are the step-by-step approach we should follow for the study?
Sure. So first part for estimation of any leachable or performing study of any leachable, first is to define the scope and the study design. So the objective should be identify, quantify, and evaluate leachables from the components like vials, stoppers, syringes, and IV bags into the drug product. So for that, we have to consider and we have to look for the container closure system components, its dosage form, for example, solutions, suspenser, or biologicals, route of administration, whether it is a parenteral inhalation, and duration and the condition of storage. So for that, reference is available FD container closure guidance as a USP chapter 1664. The second point that we have to consider is select representative sample. So as we have already discussed for the leachable, we have to mimic the real case scenario. So use the production equivalent or the commercial product batches for the evaluation. And also we can include the worst case packaging configuration uh, and consider the different lots for the packaging components to account for the variability. Then define the storage condition and duration. Store the drug product in its final container system under the real time, real temperature conditions as per the shelf life of drug. Isolated conditions can be performed to generate a supportive data, and sampling time points often includes initial t is equal to 0 for the comparison, then intermediate for example 3 months or maybe a 6 months, and end of the shelf life for example 12 months or 24 months. The study should be cover the entire shelf life of the product for the exact estimation of the leachable and its toxicological evaluation. Then after selecting all these parameters, sample preparation and extraction is the next step. Take a aliquots of the drug product and at a specified time points. Use validated extraction and sample preparation techniques, for example, liquid-liquid extraction, solid phase extraction, or direct injection can be performed. After that, preparation of the samples, analytical testings of the leachable can be done using a validated methods. And for the volatile or semi-volatile compounds, GCMS analysis should be done. LCMS for the non-volatile polar impurities, and ICP MS OES for the elemental leachables, example heavy metals. Then next step is quantify. To quantify any leachables above the AET, AET is derived from the SET value and based on the product dose. Then after analysis, the step is identification of the unknown. For that, we have to perform the mass spectral interpretation and comparison with the libraries, or we can use the analytical standard for the exit estimation. Then consider tentative identification and structure duration if not match found. After that, identifying unknowns, the next step is toxicological risk assessment. Assess any identified leachables that expected AET are of the concern. Then conduct toxicological safety evaluation using either threshold of the toxicological concern, or we can use permitted daily exposure or mutagenic impurities ICH M7 guideline for the further evaluation of the toxicological compounds and their risk.
So, Mr. Ketal, you use two terms, AET and ACT. So, can you give more information about what is AET and what is ACT?
ACT and AET we will take in the next part because it is a big terminology and it is required for the identification and evaluation of the impurities. So from ACT we can derive the AET value, and from the AET we have to calculate our LLOQ value. So AET is analytical threshold concern, and for the ACT it is a value that can be useful for the estimation of the AET value.
So, thank you. So, we will discuss these two terminology in the later part of the webinar. My next question is, what are the common compound used as a reference standard for the calibration curve for the VOC specifically?
Okay. So common compounds used as a reference standard for the calibration curve in VOI analysis can be chosen as per the USP 467 residual solvent list and VOC mixers for the commercial suppliers. So if we see the class 1 solvents, generally we have to avoid these solvents and used as a control for the detection. So benzene related compounds, that are the class 1 compounds, and here the list of compounds are presented. Class 2 solvents, they are of the limited use as a calibration standards, and here the list of all the class 2 solvents are presented. For example, methanol, toluene, and acetonitrile. Class 3 solvents are having a low toxicity and often used in the formulations. For example, ethanol, acetonitrile, isopropanol, and etc. So other common VOCs in extractable leachable calibration mixers are presented here. For example, styrene for the polystyrene materials.
Okay. So my next question is, can you please provide some practical example to understand it better?
Definitely. Here the first example is provided for the IV bags. They are the example of the PVCs and polyphenes. So common materials that are present in the IV bags are DEHP, plasticide PVC, and polyphenes. So here the list of VOC of concerns are mentioned, and the calibration mix recommended are EPA VOC mix. For example, Supelco VOC mix 1, and also we can add styrene, DEHP, related volatiles, and cyclohexane as an additional calibration tester for the evaluation of this kind of VOC leachables. The next examples is for the syringes and prefilled syringes. It is an example of glass, COP, and elastomers. and common material that can be present as a cyclic olefin polymers, rubber plungers, and lubricants. So here the list of VOC of concerns are mentioned. And for the estimation calibration mix recommended are EPA 502 524 VOC mix, and also as an additive we can add DMA, MEK, and lubricant related VOCs for the estimation of the leachables. So as an inhalation devices like MDI and DPIA, the common materials are plastic elastomers, coatings, and polyenes. And here the list of VOC of concerns are provided. The calibration mix recommended are the custom VOC mix, including chlorinated VOCs, and also we can add formulated propellants and rated impurities. For the tubing containing silicone, PVC, and TPE, the common materials are silicone elastomers, plasticide PVC, thermoplastic elastomers. Here the list of VOC of concerns are provided, and calibration mix recommended are EPA VOC mix 1 or 3, and also we can add the plasticide related VOCs, THF, and MEK. For the rubber closure and stoppers, the common materials are butyl rubber, natural rubber, curing agents, and here the list of VOC of concerns are provided. And the calibration mix recommended are VOC mix with the aromatics plus sulfur VOCs. We can also add carbon disulfide rubber specific VOCs. For the medical devices and implants, the common materials are polymers, metals with the coating and adhesives. So here the list of VOC of concerns are provided, and the calibration mix recommended are custom VOCs mix with the acrylates, chlorinated solvents, and we can also add MMA, IPA derivatives and coating related to the VOCs.
All right. So, just like the bioanalysis, do we also require to use the internal standard? And what are the compounds which are generally used as an internal standard?
Yes, internal standard is really required to avoid any variability during the sample extraction or the further step. So generally, we are calculating the response ratio. So we can use toluene D8, 1,4-dioxane D8 as an internal standard.
Okay, great. So, what are the common compounds used as a reference standard for the calibration curves for the SVOC?
Okay, we have already covered the VOCs. Now we are moving to the SVOCs, calibration mixers. So generally, we have different kind of calibration mix. So phthalates are one example as a plasticizer. So like we can use DEP, DBP, DEHP, and DBP as a plasticizer phthalates. So typical compounds included in SVOC calibration mixers are polycyclic aromatic hydrocarbons. Here the examples are provided such as naphthalene and other compounds. Nitrosamines, they are toxic and sometimes can be considered as leachables. So NDMA and NDA are the best examples. Phenols and chlorinated phenols, they are phenols and 2,4 dinitrophenols and pentachlorophenols. Anilines and aromatic amines, for example, aniline, 2-nitroaniline, and 4-chloroaniline, and others are like bisphenol, diphenylamine, hexachlorobenzene, 1,2,4, trichlorobenzene are the example of SVOC calibrations. So now we are moving to the applications in the ELL studies. So plastic and elastomers, they are the example of phthalates and BPA. Inks, adhesive and coating are the example of aromatic amines and PAHs. Rubber and stoppers, they are having nitrosamines and chlorinated phenols. Syringes and containers having SVOCs from antioxidants and UV stabilizers. So same as VOCs, we have also a internal standard for the SVOCs like naphthalene D8, phenanthrene D10, acinafin D10, and chrysine D12 as an internal standard.
Okay, great. So after the volatile and the semi-volatile organic compound calibration standards, we would like to know more about the non-volatile organic compounds. So NVOCs, if you can give example of the reference standard for the calibration curve for specifically for the NVOC.
Okay, so for the NVOCs here, tabular format is provided for the classification as well as the example of the compound. So, like antioxidants, we have BEH and other compounds. Here also a tabular format provided for the CCS and the possible leachables, comparing product type versus VOCs to be included. So here the list of commonly used internal standards for the NVOCs LCMS analysis is provided. The tabular format as a compound versus the its use.
Okay, so after that, we would like to know about the elemental impurities. So can you give more example of the reference standard for the calibration curve for the elemental impurities?
Okay, here tabular format provided for the class, then elements and the reason why we have to choose, and also we can refer the ICH to 3D USP 232 elemental impurities for the estimation of the leachables. Here a list of internal standards that can be used for the ICP-MS has been provided.
Great. So now we understood about the validation parameter, how the validation should be done, and what are the key component and the steps involved in the study of leachables. So in short, now we identified, I develop the method, validate the method, and we know about the steps involved in the study sample analysis. Now we want to interpret. So in the next webinar, we are going to focus more on interpretation, how we can interpret the levels of the different materials and the component and the chemical entities which are present in the formulation, and how it can harm the body. So our next webinar is coming up, which is focusing on the extractable and the leachable interpretation. So we will be see you at that time. Thank you so much for your time.